My name is Kwabena Bediako, and I'm presenting on response of groundnut genotypes to pre-harvest aflatoxin contamination in Ghana. Groundnut is an important grain legume in Ghana, and in fact, it constitutes a substantial proportion of the menu of many Ghanaians. However, the crop is predominantly grown in areas with a characteristic end-of-season drought, often resulting in high aflatoxin contamination in groundnuts. It is therefore important to cultivate groundnut varieties with reduced flavus infection and aflatoxin contamination. It is against this background that this study sought to identify groundnut varieties or genotypes with reduced flavus infection and aflatoxin contamination in the field. The experiment was conducted at Fumesua in the Deciduous Forest zone and at Nyankpala in the Guinea Savanna agroecological zone. The experimental materials used were 22 groundnut genotypes from ICRISAT and 5 varieties from Crops Research Institute. The experiment was laid out in a 9x3 alpha lattice with 3 replications. The planting at both locations was delayed in order to expose the genotypes to 30 days terminal drought. And again, a local aflatogenic flavus strain was inoculated onto the plants 60 days after planting. These are conditions required to ensure flavus infection in the field and subsequent aflatoxin contamination. We collected data on kernel infection by flavus, aflatoxin concentration, and also on yield. Data were subjected to analysis of variance using SARS. Place 1 and 2 show genotypes with reduced flavus load and genotypes with high flavus load 7 days after incubation. From Table 1, genotypes ICG 4729, ICGB 03401, ICGB 03331 had aflatoxin levels below 20 nanograms per gram, and that is the permissible level set by the Ghana Standards Authority and the United States Food and Drug Administration. Compared to the most popular variety we have in Ghana, which is Chinese, it had aflatoxin levels more than the 20 nanograms per gram. The genotypes with low aflatoxin levels also had low flavus load from Table 2. And this means that these genotypes will be used as parents to develop aflatoxin resistant varieties. From Figure 1, genotypes ICGB 03331 and ICGB 03401 also had pod yield and home yield similar to or higher than Chinese, which means that these genotypes could be good varieties in Ghana when they are evaluated at multi-locations and their performance established. In conclusion, we found significant genetic variation among groundnut genotypes for aflatoxin contamination and seed infection by flavus, and also for yield. And this indicates that there is possible selection for improvement of these traits. Genotypes ICGB 0331 and ICGB 03401 consistently showed low aflatoxin levels and low to moderate seed infection by flavus. Again, these genotypes also had appreciable pod and home yield and could serve as potential varieties in Ghana, following further evaluation at multiple locations. Thank you.